Hello, la la la. For all you Seinfeld fans out there. Hi, Mrs. Smith.
Hi, Mr. Suitor. You're so far away.
Who's here today? Aiden in the house. Deanna. Hallie. Nelvik.
Maddie, Morgan,
Maddie,
Libby, Lauren.
Moth. Libby. Lauren. Who else? Harley Collins. Huh, Harley, nice to see you. I haven't seen you in so long. Come back. Grace. Who else? Manny.
Yasmina. Yazzy's here. Kenzie's here. Keeney's here.
Keeney. The house.
Emma, Rosa.
Who's Jax Dell?
Jaden.
That's a new one for me.
Jackson.
And Yo Boy Crazy Cray is a new one for me too.
Caden. Because it's Caden.
Tyler. Ethan.
Carson. Seeing some new names. That's good.
Yeah, Caden, by the way. Good. Yeah, new names are good, and we have a way that we're going to kind of keep a better tally on who's here today, because we did make a promise to you that if you came to these, that it would positively affect your overall grade for fourth marking period, and
Correct.
We have a way to do that. But you're going to have to hang on a little bit to find out what that way is. Jaden, Emma, Maya.
Anybody we missed?
Did we miss anybody?
I don't think so.
I hope not. All right, so today we are reviewing Keystone. This is it. This is how we're going to try and review. That gives us like 7 Fridays, I think, to go over stuff before you take this ridiculous test.
So there's two things you need. This.
Yes, 2 things.
And this. Oh, and
so maybe we're not the best at telling you this prior to this today, but if you have a blank piece of paper and a writing utensil, it would be really helpful for you. It might help you earn some extra points this marking period. So if you don't have an extra piece of paper and a writing utensil, grab one now.
We'll stall for a minute.
And then if you don't, you might just have to come back and do it a little bit, but it won't be that big of a deal. All right. So from now on, writing utensil, piece of paper, answer sheet. What else? That it? Good. Mrs. Smith.
Yes. That is me.
Basic biological principles. Assessment anchor number 1 of the biology keystone.
Correct.
Show them what they need on your little camera there.
Great. All right, so we're going to take our blank piece of paper. Ta-da. And we're going to create a really quick graphic organizer. So a pretty famous one to start off with is our good old Venn diagram. And I want to say, as I am drawing this, to remind you all that when you take the keystones, there is absolutely no time limit, which means everything that we will practice, that we will do, that we will go over, you can practice making these. You can walk into the day of the keystone and you can recreate these on the scrap paper. You know the scrap paper that you get, that you've never touched, that you never do anything with? You can essentially make yourself an answer key for the keystone if you practice, because you have as much time as you want. So all the things that we are going to show you are really helpful to study beforehand, and they're also really helpful to actually reproduce and use when you are on or taking the Keystone exam. So today, like I said, we're going over some of the things from the very beginning of the year with basic biological principles. And so that particular unit talks a lot about the different types of cells. And so we're going to look at the difference today between prokaryotes and hopefully we know the other one that I'm going to put on the other side. Prokaryotes versus, see if anybody can beat me writing. Prokaryotes and...
So is that kind of like you can't take a cheat sheet into the keystones, but nothing can stop you from going in there and making it after you get in the keystones?
Exactly.
Yeah.
Nobody's got it in the chat? Oh, there we go.
Yo, boy, Crazy Cray.
All right, so we're talking about prokaryotes versus eukaryotes, or prokaryotic versus eukaryotic cells. So the first thing we want to establish is, what are both of these? And so a lot of times what we think of first when we think about prokaryotes versus eukaryotes is that we have a single-celled or unicellular organism when we're talking about prokaryotes, and eukaryotes are a multicellular organism. So we talk about prokaryotes versus eukaryotes. A lot of times we think single cell versus multi-cell, but I want to make sure that when we talk about this, really prokaryotes, a lot of times we're talking about as bacteria, but you can actually have single-celled eukaryotic cells, because it's not just our plant and animal cell, but it's also our protists and our fungi. And so you can have some single-celled protists that are eukaryotic cells. Okay, so, but overall, typically we talk about a unicellular and a multicellular organism. Okay, so when we talk about this, something else that we want to kind of discuss, it's a big one, is that in a prokaryotic cell, there are no membrane-bound organelles. Eukaryotic, membrane-bound organelles. Like what? Can in the chat as I'm writing, somebody tell me what a membrane-bound organelle is?
Lots of examples to choose from, right? Is that what you're looking for, Mrs. Smith?
Yes, lots to choose from. So Emma's got mitochondria, great.
Libya ribosome, nice.
Yes.
But is a ribosome a membrane-bound organelle? No. Sorry, Libby.
So I got nucleus, mitochondria.
Is really just a piece of RNA. It's the pirate RNA. It's the rRNA, actually, is the ribosome.
And then I'll just throw in here Golgi apparatus. Oh, good. Somebody said ER. Okay, so there's a bunch to choose from. So as far as ribosomes go, like Mr. Suter said, ribosomes are actually just a piece of RNA, specific RNA. And so ribosomes is actually something that they both have. It's the only really quote unquote organelle that is in prokaryotes and eukaryotes. So in prokaryotic cells, when we say there's no membrane-bound organelle, that means there's no nucleus. So instead, what we have is this nucleoid area, which I think I'm spelling that right off the top of my head. I'm going to put like area in quotes, because that's where all the genetic information is, which in a eukaryotic cell, it's going to be contained within the nucleus. So both of them do have DNA or genetic material. They all need that in order to reproduce. So they all have DNA, it's just where it is that makes a difference. And then as far as reproduction, multicellular organisms like eukaryotes are going to go through reproduction in the form of something called mitosis, which hopefully sounds familiar and is upcoming in our units, where a single cell is going to reproduce in something called binary fission, where they actually just separate into a clone. And so binary fission creates kind of cloned copies, where with mitosis and then also technically meiosis, it can be a little bit different. So again, we have ribosomes and genetic material that they both have. I saw somebody else in the chat write down cytoplasm. So cytoplasm isn't actually a membrane-bound organelle. It's all of the kind of liquidy substance that is in the cell. It's mainly comprised of water, and so every cell has cytoplasm. So every cell has cytoplasm, has ribosomes, has some sort of genetic material. And then there is, of course, the thing that holds it all in together, which I tell my students when we talk about, like, we draw a cell, we draw a smiley face at the beginning of that unit, you always drew the membrane first. So the cell membrane or the plasma membrane. Did I miss anything, Mr. Suter?
Negative.
Okay, so what we did here quickly is, and this even took more time because I walked through it, but if you were to walk into the Keystone exam and grab a quick piece of paper, create this, and whether or not you drew the circles, whether or not you just draw a chart and write down prokaryotes and eukaryotes, you can even flip it over and say animal and plant cells, I know the difference between those, and you create that for yourself. Then when you go to answer questions, like Mr. Suter is going to go over in a second, you already pretty much have the answers there in front of you. Mm-hmm. So Mr. Suter, do you want to flip over to those questions? And I'm going to leave this up and kind of push it off, you know, if you want to make that small off to the side that we can come back to if we're looking at a question.
So two things. One thing, before we switch gears, hello, welcome to Get Some Extra Points in Biology Time. In the chat, you are about to see a link pop up to a Google form. If you click on that Google form real quick, please pick the correct period. Please pick the correct teacher. I'm Mr. Suter. That is Mrs. Smith. And fill out that form. You'll be able to fill that form out anytime between now and I think 11:15.
Yeah, let me finish. You should do this now. Now remember, I have classed 1st, 2nd, and 7th. So you don't have me 6th or AB or DE or any of those other periods. If you have me, it's 1st, 2nd, and 7th.
If you pick the wrong period, you might not get as many points as you want. And so we are going to collect these. We're going to do these every week. And at the end of the review, we will go through, see how many times that you were a party to this, how many times you came, and give you bonus points accordingly. So do not expect bonus points to be showing up every week. They will all come at the end, along with points for completing the study guide and having the graphic organizers that we do doing this stapled to them. So there's some work, but we're 12 minutes in, 13 minutes in. Let's get into those things.
Questions.
You did, and let's see how you did. And now I'm just talking while I'm trying to figure out how to share my screen. Share screen. Let's do a window here, and we're gonna do not that window, but that window. Share. Boom. All right. So question number 1 that you were to answer throughout this week is: which characteristic is shared by all prokaryotic cells and eukaryotic cells? And so whenever these Keystone questions are hard if you don't think about them. They're not hard if you take some time to kind of break them down and think about them a little bit. Whenever you get one of the questions, the first thing you need to do, and this is probably an easier one, but the first thing you need to do is really think about, what is it asking you? What do all prokaryotic cells and all eukaryotic cells have in common? Go back to her thing. Right there it is. Cytoplasm, ribosomes, DNA, cell membrane. So let's go back. Yeah, Grace, we can put it back up at the end. Go back. What are our choices? A, the ability to store hereditary information. What is hereditary information? That's DNA, right? DNA carries the things that make you you. The use of organelles to control cell processes. The use of cellular respiration for energy release. Ability to move and respond to environmental stimuli. I'm going to pull you back up, Mrs. Smith. that cell respiration thing, you need to have a mitochondria for that. They don't have it. So the only thing that this Venn diagram shows us that they have in place.
Ding, ding, right there in center.
Yep. All right. Back to this. Question number 2. Living organisms can be classified as prokaryotes or eukaryotes. Which 2 structures are common to both prokaryotic and eukaryotic cells? Jumping back to Mrs. Smith's wonderful graphic organizer. Cytoplasm, ribosomes, DNA, cell membrane. Those are the 4 things that are common. So when we look at these answers, cell wall and nucleus?
Negative.
Cell wall maybe, but nucleus, no way. Cell wall and chloroplast? Chloroplast is a membrane-bound organelle, no way. Plasma, membrane, and a nucleus? Nucleus is a membrane-bound organelle, no way. Plasma, membrane, and cytoplasm.
And it's okay to write on the test itself when you have the keystone in front of you.
Yeah,
if you're sure it's not it, cross it out.
Right. And if I had a pen right now, I would be crossing out nucleus, chloroplast, and nucleus because I know those are membrane-bound organelles. What was number 1 again? Number 1, the answer was
DNA, hereditary information.
DNA, hereditary information. And see, that's the thing. Because we've been talking about DNA. You would not have missed this if it just said DNA. But people miss it because it says hereditary information. So you really have to think about what you're reading on these. All right, coming down to number 3. Prokaryotic cells are generally much smaller than eukaryotic cells. They're just telling you that. Like, hey, duh, you knew that, right? So identify a structural difference between prokaryotic cells and eukaryotic cells that is directly related to a difference in their size. So in the chat, why does a eukaryotic cell have to be bigger than a prokaryotic cell? Putting the graphic organizer up again. In the chat, give me 3 words that really drive this home. In the chat, please take part. Membrane, yeah, Brian, on it. Membrane-bound organelles, right? There's parts in there. It has to be bigger to store all those parts. Or is it bigger because it has, or does it have the parts because it's bigger? Kind of talked about that. It's the chicken or the egg. Which came first, right? Once the cell gets to a certain size, it has to be organized so it can move things around, so it can bring food in and get energy from one place to another and all that good stuff. So is the organization what allows for the size? Yeah. Let's be more efficient. More efficient. Very good. Look at you with your little marker over there, Mrs. Smith. You're awesome. All right. Next part of that question. Based on the structural difference, explain why a prokaryotic cell can be much smaller than a eukaryotic cell. Again, go back to this. Why can it be so much smaller? No membrane-bound organelles. In fact, I would argue it has to be smaller because it's not as organized. How does it move things around? How does it move food around? How does it move proteins around? You know, if you are too big, right? Think of little communities like when the settlers first started going out west and would first settle something. Their community was really small and probably really close together, right? Like everybody lived in a really kind of small area and helped each other out. But when you get to like a big city, now you need fire departments and you need, you know, hospitals and you need, you know, the water people or whoever, the trash collectors. And it needs to be much more complex as the city gets bigger. Same with the cell. As the cell got bigger, it needs to be more complex. All right. Last essay question part. Describe one similarity between a prokaryotic cell and a eukaryotic cell that's independent of size. Boy, I feel like we're beating this to death today, but there it is.
Take your pick.
Right. Cytoplasm. The stuff inside doesn't matter how big the cell is. Ribosomes doesn't matter. DNA doesn't matter. Cell membrane. Doesn't matter. Mr. Smith, you want to do the last one?
Sure.
That was the last one. I'll read it. This is what I love about the Keystones, because when I first read this, I thought it was an unfair question because we never talked about it. And we probably haven't talked about it enough this year, so now's a good time. Alveoli are microscopic air sacs in the lungs of mammals. Which statement best describes the structure of alveoli, of how the structure of alveoli allows the lungs to function properly?
So before you even get overwhelmed with maybe you don't know what alveoli are, hopefully you know what lungs are. And what do lungs do? What's the purpose of your lungs? What is the purpose of your lungs? Okay, so getting oxygen, okay? So part of it is getting oxygen to allow you to breathe, all of that stuff. Great. So when I look at this, the first thing I'm going to do is to try and, look at questions or answers in there that talk about actually breathing. And Mr. Shooter just like pulled it away, and so I can't actually.
I'm sorry. I'm coming back.
And so, I look at letter B and it says flexibility of lungs. Well, that really doesn't help me to function properly as far as what lungs do. So I'd get rid of that one right away. Okay. And then if I'm talking about amount of energy transferred from lungs to the blood in A, well, that's not the purpose of lungs. So I've just cut it down to a 50-50 shot pretty quickly from just knowing what lungs do. And so C says increase the volume of lungs, allowing more oxygen to be inhaled. And then D says they increase the surface area of the lungs, allowing efficient gas exchange. And so when we talk about gas exchange, we're talking about not only bringing oxygen in, but also releasing carbon dioxide back to go out of the body. So that really is what lungs are for gas exchange. And when I see that surface area, that makes a big key impact on me because I know surface area is a big important part of biology. So D would be the best choice for that one.
So real quick, because I don't know how Mrs. Smith maybe brought up surface area more than I have, but I haven't brought it up enough. Let's talk about just real quick. Let me give you 3 or 4 quick examples of why surface area is so important. First of all, if I asked you what melts faster, a block of ice or a bunch of little, or if we take the block and we break it into small pieces, you'd say the small pieces. Why? Because the air can hit more of those pieces. What burns faster? A huge log or a log that's been cut up into pieces. Why? When we think about even cellular respiration, when we talk about glycolysis, that starts cellular respiration, right? What's the first thing we did with a glucose molecule? We broke it down into smaller pieces before we sent it into mitochondria. Let me give you 2 more examples on my little whiteboard here. If I were to draw, can I draw a circle? Here's a circle. This is your intestine. Oh, this is a good one, right? This is your intestine. This is like I reached into your body, pulled your large intestine out, cut it, and now we're peering into it, right? So, don't hold your eyeball too close kind of thing, right? You get brown. Never mind. You're looking into your intestine. What you would see inside, and this, excuse me, anatomy teacher, if this is terrible, but we get the point across, right? You would see all these little finger-like projections and hard to draw coming into your intestine. Mrs. Smith would tell you the proper word for those finger-like projections. Starts with a P, right? If Mrs. Smith is listening. Did I lose you?
No, I'm here. I'm just laughing. Go ahead. I
What's
was
the
trying not
proper word
to.
for the finger-like projections, Mrs. Smith?
Within the intestines?
Yeah, they'd be it's a, I forget.
The villi?
Yeah, maybe.
Okay.
Anyway, so what's happening in your intestine is that we're increasing surface area, which actually kind of interesting. Like if you were to draw a mitochondria, remember how we draw that second membrane in the mitochondria? And I think we did talk about this when we did this before, right? The second membrane had, what did we call those? Oh, bonus question of the day. What were those little finger-like projections in the mitochondria called?
Starts with a C.
But the point here is all this is to increase surface area, right? Because the more area that's touching what's happening, the more exchange can take place. So if you go back to that question and we had a lung, how's that for my lung, Mrs. Smith?
Awesome.
The one question said about it increases volume. Well, you don't increase the volume if you fill it with a bunch of little sacs, right? These sacs aren't increasing the volume. They're actually decreasing the volume. But everywhere those sacs are touching air, they're increasing the surface area, if that makes sense. Hopefully that makes sense.
Great job.
Why are you laughing at me? You're such a... you want to get out of here? I love Mrs. Smith. All right, so... Did we get it all?
I think we got it all. The game. The game. The review game.
So at the bottom of the Keystone review sheet that is posted in Moodle,
It says Keystone Review Guide.
here's a link to that guide. At the bottom of that guide is a link to...
And yes, it is called Chris Day. Very good.
Who got it?
One of your students.
Which one? Oh, yes, Miguel. Very nice. At the bottom of that page, I could have given better. I just got nervous because Mrs. Smith started laughing at me.
I'm sorry. You said it started with a P, and I'm like, what are you trying to think of there? So, PV, I guess, you know.
It doesn't start with P?
No, villi, V-I-L-L-I.
I thought there was, yeah, see? I'm stupid.
Are you talking about the papillae that's on your tongue?
No, I mean, I don't know. I haven't. Drop it.
Okay.
You're so mean. Look here. Here it is. Basic biological principles. Game code. There's a game code there. Ends April 11th on Sunday. We want you sometime today, tomorrow, or Sunday to play the game. There's a bunch of questions there, but it goes over basic biological principle stuff. This is the first game. We'll see. We might be taking track of who wins each game as well. So please make sure you put in your real name. Don't put, if you put in like some, like, I don't know, Crazy Cray boy, I might not know who it is. Sorry, Kaden. I love it. You never change. All right. Anything else?
Nope. Just remember that I know we put the link in the chat, but it's also in both of our Moodles at the top. So you can find it again. That's where there's a link to the review sheet, everything that you need. There's also some really good review videos and slide decks and things like that that you can go through for additional help in each of the subject areas that are in there. So the choice is yours.
Yeah, I mean, I got to tell you, we have more people in here right now than maybe on a typical Friday, but not enough on here right now to make me feel good about your chances of passing, most people's chances of passing the Keystone. You know what, Mrs. Smith, it's a weird year. I get it. It's a weird year with everything happening with COVID. I understand there's a lot of, uncertainty and a lot of people that at some point in time, though, guys, you got to stop making excuses and start climbing over some of these hurdles that are put in front of you. Like, yeah, you might have a great excuse to fail bio this year, but next year you're going to be sitting in a class with a bunch of freshmen. And I'm not talking to those of you in this room probably right now, but maybe you could pass it on to your friends. All right, Morgan. So if you're done, you're out. Morgan, if you have an off-topic question, you can shoot real quick, but everybody else can get out.
11:01. We reviewed a whole unit in 30 minutes.
Yeah. Paywalled by Suter. I'm not sure I'm familiar with that term. Is that a sign of age, Mr. Smith?
What, huh?
Is that a sign of age?
What? What'd you say?
Villi is what I said.
Oh, tomorrow is a birthday. But if we play the monkey song, then we might get
No, I don't care.
yelled at by YouTube.
Tomorrow is your birthday? Well, if that's the case.
Bum. Come on, Sudi, you have to be faster than that.
I can only go as fast as my computer goes. If that's the case, birthday.
I didn't even get the monkey song on my birthday.
Oh my gosh. Da-da-da-da-da-da.
This is ridiculous.
Where is it? Come on. Why is
Contact
it
free services.
an ad? Copyright for playing somebody's ad on your channel? Oh, I don't know if we had sound set up right.
But you're not playing it. It's playing not on the screen. Share it. Share the screen, Stuart.
Oh. All right. I so must not do that without it being in class with sixth period, because sixth period when we play that, it's turning into like a scene out of High School Musical. That's all I have to say. It's like there's dancing and singing, and it's great. Have a good day, everybody. Peace.